So in the wonderful land of Penguinland, one of the aspiring scientists called Lizzie was interested about how two traits inherit from one generation to each other, and she wanted to test out the Mendel's second law on Drosophilus, the fruit flies. This law is also known as the law of independent assortment. Here are the fruit flies, and she decided to observe two traits, the color of an eye and the color of the body. She also concluded prior to the test that the white color and the yellowish body is recessive traits. Well, here her and here the where that's when her experiments begin. So she decides to cross the heterozygous individual and completely recessive individuals to each other, and she observed very interesting scenario. There were lots of parental phenotypes, but there were so small amounts of recombinant phenotypes. In classical Mendelian inheritance, we would get 1 to 1 to 1 to 1 ratio, but it didn't happen. Why? Is the law of independent assortment false? Well, it's really early to make conclusions, and law of independent assortment only works in certain cases, but it is not incorrect. First of all, we need to discuss how germ cells actually work and actually get created. These germ cells, called gametes, after fertilization merge to each other and become the zygote from which offspring generates. So here we have the germ cell precursor from which the germ cell, the haploid cell, gets generated. As you can see, if the two genes marked with these boxes are on the separate chromosomes, in germ cell they will also get inherited separately. As such, they would assort independently. But if both genes are on the same chromosome, they would not assort independently and they would inherit together, meaning that if we had some allele combination of genes on the same chromosomes, they would not change and get inherited as it is. But by that we would only get the parental phenotypes we mentioned earlier. So how did we get recombinant phenotypes? Well, we need to explain meiosis. Meiosis will be explained in detail in the future videos, but let's quickly sum it up. So, at the start of the meiosis, the two homologous chromosomes with two chromatids, here are the genes again, are merged, not merged, and they have overlapping areas, and they create a bivalent. This is called a bivalent formation, this process. Now what they can do is actually switch their genetic material. So at the homologous parts of their chromosomes, they would switch each other's parts, and as such this process is called crossing over. And after the meiosis 1, we would get a segregated chromosomes with different genetic materials from other homologous chromosomes too, meaning that the allele combination we had before has been changed. As such, we have ability to generate recombinant offspring. Now, from these techniques, by crossing the heterozygous individual to completely recessive individual, we can create the genetic maps. So what are the genetic maps? Genetic maps tell us the approximate location of the genes on the chromosome. Now, this is not the physical markers, the actual physical markers, but the genetic maps are created by conducting our crosses mentioned earlier. So let's say the distance between these two genes are x. The x will be calculated by a over b times 100, and it is measured in centimorgans. Well, let's define what are A and B. A is the amount of recombinant offspring and B is the amount of total offspring. So, let's think for a second. If A is the amount of recombinant offspring, it means that as far as genes are, there are more space between them for crossing over to happen. Meaning that amount of recombinant offspring will rise. As such, we need to find what is the ratio between the recombinant offspring and the total offspring. And for the classical cases, it will be 50%. For the classical cases, because they would get generated by similar means. But when they are linked, as such, meaning they are on the same chromosome, this A is a lot smaller. As such, we can get some approximate value, and we multiply it on the 100 because we're measuring in Morgan centi, meaning the hundredth. Now, our scientist has drawn some conclusions. So first of all, some genes are linked to each other and are inherited at the same time, as mentioned for the Drosophila, and the actual scientist, Morgan, demonstrated that in his experiments. The recombinant offspring are created due to crossing over in meiosis. This was when the homologous parts of the same chromosome or same homologous chromosomes were switched over. As such, we would get the different allele combinations. And the cross between a deheterozygous and a complete recessive individual can be used to create the genetic maps, aka sequence of how genes are located on the chromosome. Thanks for watching.